have yeah, some wonderful people here that will be speaking tonight. I'm just going to do our introduction. I'm Alora. I'm one of our project coordinators, and then Anne is in here too. She'll be kind of helping with Zoom stuff until she jumps in for the protocol overview later on. So yeah, thanks for coming. Thanks for being here, and we'll just get started. I think that was all I was going to say at the beginning. Yeah, stay on mute. You're being recorded. That's good. Yeah? Okay. I just wait for Anne to give me the thumbs up and I know that everything is good. All right. So yeah, wanted to start tonight by with a land acknowledgement. So I'm on Lekwungen territory today. I know lots of us are. I spend most of my time on Lekwungen and Wasanich territory, and it's really cool because we probably have folks from all different corners of the province here, or at least I'm hoping. In a minute or two, we'll actually do a little poll where you can answer where you're from, and we can kind of see where folks are dispersed. But wanted to touch on this because I think for me, and for many of us, being interested in fungi and biodiversity goes or is a bit more broad than that. Like for me, when I go out in the forest, I'm crawling around on the forest floor. I'm noticing all these little things that I normally never would notice. And it's really been like an avenue for feeling more connected with the ecosystem around me, with the land that I live on. And I'm hoping that MycoMap can also be a avenue for that for other people. And that when we go out in the forest, we can not only, be aware of like how to respectfully move through the forest, respectfully interact with fungi, but how we can sort of build relationships and interactions that are going to be contributing to systems that are affirming of life, human and otherwise. So wanted to just start with that. And yeah, I'm excited to get into it. So, first thing here, everyone probably knows what this project is, or at least has a general idea since you made it to this meeting. So really what MycoMap BC is a community science effort to document fungal biodiversity in BC. And it's really great that all of you are here because even though we have some people in this meeting that do make tons of collections, are really good like power collectors, the more people we have collecting and going out there and kind of knowing how to participate in this process, the more success we'll have, the more data we'll have, the more impactful this project will be. So Anne is going to get into the nitty-gritty later, really, of like how to participate, what do you need to do, how to get all set up to participate in the project. So I'll leave that to her and just kind of keep going with our intro here, but yeah, we'll get to that a little bit later. Okay, polls. Let's do our polls. I've never done polls on Zoom before, but I think they're really fun. I actually am dependent on Anne doing them here for me. So Anne, if we could first do the one that's like, where are you from? Yeah. So where are you joining from? I put some arbitrary groupings of places in BC for people to vote for. And we'll give it like 30 seconds here for everyone to vote if they'd like. Okay, looks like most people have voted. Last chance here. And everyone can see the results live, right? That's not just me. If someone could give a thumbs up emoji.
I don't see anything, Ann or Elora.
Oh.
Oh, there it is.
There. I just have to share the results once they're in.
Oh, okay. Beautiful. Okay, so a little unsurprising, I guess, that we have a big chunk of people from Vancouver Island since that's where I guess the project originated. Some people from the Lower Mainland, and only the Sunshine Coast is unrepresented. But it's great to see some people from, wow, even some people from outside of BC. Cool. Well, we can end that one. And then I'll exit that. Two more questions just to gauge kind of like what everyone's experience is in the world of mycology. Do you want to pull up one of those? Yeah. So just whatever you think is the best fit here. Interested in like how experienced folks are and how you found yourself at this meeting. Well, I get to watch the little things move live, which is pretty fun. But I guess Anne has to share the results. Almost everyone's voted. Last chance to vote.
Okay, we'll call it.
Yeah, we'll call it. Okay, wow. Actually, it's really exciting to me that there are the biggest chunk of people here have chosen beginner, which is really cool because I guess when I first started, I wasn't really making collections yet. It took me a couple years of being in the like myco world to get into the groove of making collections. So that's cool. It's fun to have lots of beginners here. I think there's lots of ways this project can lend itself to like learning the basics of mycology, which is great. Sweet. And then we can do our last poll, which is about
Hang
collection.
on.
Yeah, no
One.
problem.
There we go.
I'm like, and the poll. You're doing
Technical
great.
difficulties.
Yeah. Okay, it says, have you collected fungi for vouchering and sequencing before?
Oh man, I'm loving these results.
Okay. Oh yeah, I spelled Kootenays wrong. Nice. Yep, definitely meant Kootenays the right way. Cool. Wow. Okay. So yeah, the biggest majority of people have not ever collected fungi before. That's great because we're going to teach you how, and it's really not all that hard. So that's fantastic. And yeah, some people who have lots of times, wonderful. Oh, are the results shared now?
Yes, they should be.
Okay, great.
I'll stop.
Great.
Okay.
Sweet. Okay. Well, the last thing I'm gonna draw everyone's attention to before I pass it off to some other folks, or I think this is one of my last slides anyway, is our website. So if you haven't yet poked around on this web page, it's hosted on the Mycota website. So if you Google Mycota or you go to mycomapbc.org, you'll find this specific page, which has tons of great information. It really has like the core things you need to know in that it has our protocol linked, it has our iNaturalist project linked, and it has the Google form where you fill it out. It's just a simple Google form with some data collection, and then once you finish it, you get the address of where to send the specimens. So anyway, Anne will go over that, but just drawing your attention to if you have not yet found yourself on the website, there's a ton of helpful stuff. There's a bunch of like frequently asked questions and answers that you might find useful. Yeah, and it's just a good place to go if you find yourself with questions down the road. Also, we have an Instagram account. I'm hoping that as people start making collections, you can tag this account and we can share them and sort of gain traction and visibility to other people who might want to be involved. But anyway, if you're on Instagram, you can find it at MycoMap BC. Sweet. Oh yeah. So this is a big one. We are really lucky that we've had lots of groups that were excited to help support this. So we're able to pay Mycota Lab for their work, and you'll hear from Stephen of Mycota a little bit later on. But yeah, we're happy to be able to support them with these funds. So the South Vancouver Island Mycological Society, the Vancouver Mycological Society, the Society for Education, Research, and Arts on the Land, the BC Biodiversity Program, UNBC, Machosen Foundation, and Whistler Naturalists. So those are all the groups that contributed funds. And it was pretty awesome because when this project sort of started developing and I was reaching out to these different groups, like everybody was so keen to see this happen. It really didn't feel hard to find the interest to fund this project, which, yeah, was just great and made my life really easy. So thanks. And then thank you also to all the people who have helped make this happen. Myself and Anne have been sort of like coordinating things, but there are a lot of mycologists and mycology enthusiasts who have come together to kind of like shape how this would look and provide input and all that kind of stuff. So that's been really, really helpful too. And it wouldn't have, yeah, it wouldn't have come together without like a large group of people working on it together. So thanks. Okay. So in a second here, we're going to talk about why is this work? Why is this project important? Why are we bothering to do this? Kind of the why behind MycoMap. And we'll hear from Kem, Andy, and Shannon about the sequencing revolution, the status of INAT sequences in BC, and then some examples of things we might discover through this project. And yeah, I'm happy to introduce these three folks. When I joined the Mycological Society a few years ago, they're very prominent figures that I was able to learn a lot from. The first ever like multi-day workshop I went to, Shannon was running, and I knew absolutely nothing. And I've learned a couple of things since then from all of these three people. So yeah, I'm really, really happy to introduce them. I'll read out their bios here. So Cam will be going first, and Cam is a naturalist and a writer. And after retiring from a career as a college and university professor, Cam moved from his home on Ontario's Grand River to the southern tip of Vancouver Island in 2004. He soon developed an abiding passion to know the mushrooms of Western North America. Besides co-authoring Mushrooms of British Columbia, which I'm sure many of us own. I wish I could reach my bookshelf from here and show you the book. I'm sure many of us own and love this book. Cam is the author of 7 books, including Boundary Layer, and he has been secretary and speaker coordinator for the South Vancouver Island Mycological Society. Following Ken will be Andy, who's Kem's co-author, as we'll hear in a second. Andy McKinnon is a forest ecologist who lives in Metchosin, BC. He is co-author of 6 best-selling books about plants of Western North America and co-author of the Royal BC Museum Handbook, Mushrooms of British Columbia. Andy is also a dedicated conservationist. He's the board chair of Habitat Acquisition Trust, Southern Vancouver Island's regional land trust, and has been involved in the creation of protected areas on Vancouver Island and alongside BC's coast in Haida Gwaii in the Great Bear Rainforest. In August 2024, in Telluride, Colorado, Andy McKinnon received the Linkoff Award for significant contributions to American mycology. He is the president of the South Vancouver Island Mycological Society and an enthusiastic participant, speaker, and field trip leader for various mushroom festivals in southwestern BC and the US Pacific Northwest each autumn. And last but definitely not least is Shannon Birch, who is a retired soil biologist and a mycologist, as well as a founding member of the South Vancouver Island Mycological Society, for which she currently serves as secretary, and the Truffle Association of British Columbia, for which she currently serves as president. So I will head to our next slide here and just pass it off to you, Kem. And if Anne, you can spotlight Kem and take me off, we'll be good to go.
Okay, thank you, Laura. This is just a bit of a backgrounder, and then I'll hand it over to other people. I'm going to talk for just a moment about the distinction between genotype and phenotype. It's a bit more than a century old now, this distinction, and it's become fundamental to modern biology. Next slide. In brief, an organism's genotype is its hereditary information. And today we identify the genotype, at least for everything above a virus level, with the organism's DNA. An organism's phenotype, in contrast with, this is from the Greek word phino, meaning to appear or show, is the expression of its genotype under the influence of the environment. Phenotype encompasses a lot of things. It's cellular chemistry, macroscopic and microscopic morphology, developmental processes, behavior, adaptation to environment. In general, changes in the genotype result in changes to the phenotype, and not the other way around. But of course, since the breakdown of the Darwinian synthesis in the last decades of the 20th century, biologists no longer hold this as dogma. Their minds have been changed by epigenetics, horizontal gene transfer, but it's still a useful way of thinking about genotype and phenotype. The genotype expresses itself as phenotype. In a perfect world, any change in the genotype from evolutionary processes generally would result in a change in phenotype. There are, however, many ways that this can fail to happen. For example, some genetic changes result in no phenotypic changes. We have many sections of non-coding DNA in our genome, and many sections of that changes to it have no expression whatsoever. And then on the other side, some large phenotypic changes have no basis in genetic differences. The classic example would be twins. I have twin grandsons. They're identical twin grandsons. And when they were conceived, they were identical. But every day they grow more different. The mushroom example of this is probably Schizophilum commune, the split gill. It's all over the world. It's one of the most common mushrooms, and it has so many different appearances. It can look like a polypore, it can look like a gilled mushroom, and yet, as far as we know, every Schizophilum all over the world has the same genotype, but it expresses itself differently. We sometimes refer to this disconnection between genotype and phenotype as phenotypic plasticity. It can be high or it can be low. So what does this have to do with mushrooms? The problem comes in how we determine species differences in kingdom fungi. When it was difficult or impractical to directly examine the genome, we used phenotypic changes to determine when there were species level differences. Until the 1990s, phenotypic differentiation held sway. Perhaps the only exception to this were mating studies in which we presumably got some view of the genome directly. But since then, there have been a lot of changes, and the cost of sequencing has continued to fall. And here's a graph you will see in many different places. It comes out of the Human Genome Project. And it shows the fall and the cost of sequencing. And now this is a logarithmic graph. If you saw this in a non-logarithmic graph, it would look like a cliff. That is how fast the cost of sequencing has come down and continues to come down. Next slide. The economic and technical revolution in sequencing technology has changed how we think about species. Today, it's no longer possible to publish species descriptions without some reference to their genetic differences determined through sequencing. We've reached the point, in fact, where we're prepared in some special cases to differentiate species on purely genetic data. Though out of deference to several centuries of species studies with only phenotypic tools, we always try to find and include phenotypic data. Research groups based primarily in universities have led the way into this new world of genotype as part of species definition. In the last decade, however, the lower costs of sequencing have opened the door to citizen science contributions, and a number of mushroom clubs have started to include sequencing in their club activities. The MycoMap BC project is our first major step here in BC into this new world of genotypes. Andy.
Thanks, Cam. I want to talk just for a couple of minutes here about why we felt strongly it was important to mount this initiative in British Columbia specifically. And I'm going to talk a bit about INAT sequencing in British Columbia. I'm going to speak more broadly too. Next, please. BC is a remarkable place to study fungi or just about any other group of living organisms. It is 94.5 million hectares with a mere 5 million people largely clustered down into the bottom left-hand corner. And it's one of the most ecologically, climatically, and physiographically diverse areas in the world at temperate latitudes. The wettest places, the driest places, the hottest places, and some of the coldest places in Canada are in British Columbia. And as a result, we have more species of just about any other group of organism you can think of in British Columbia than any other jurisdiction in Canada. Next, please. When we're talking about diversity of organisms, it's always important, I feel, to consider that more than 50% of living organisms live below the ground. And so if we are beginning to understand the above ground community fairly well, then we might have a rudimentary grasp on somewhat less than 50% of the organisms in any ecosystem. For example, if we look at ectomycorrhizae, next please, you can see that a cubic centimeter of forest soil contains on average 1.75 kilometers of fungal hyphae, probably representing in that cubic centimeter an area of soil about the size of your pinky fingernail in 3 dimensions, 100 to 400 species. Well, 1.75 kilometers in each cubic centimeter, 100 to 400 species of ectomycorrhizal fungi in each hectare. So it's an extraordinary diversity. Next, please. And then you look at, from this remarkable paper by the people of the Society for the Protection of Underground Networks that came out just less than 2 months ago now in Nature. The left-hand graph shows, not surprisingly, that for arbuscular mycorrhizae, the temperate conifer forests are not particularly diverse. But if you look at the right-hand graph for ectomycorrhizal fungi, the temperate conifer forests are some of the most diverse globally. And of course, they dominate the landscape of British Columbia. So what we have is an ecologically incredibly diverse area dominated by these temperate conifer forests, which are remarkably diverse for ectomycorrhizal fungi. So great opportunities to learn about the amazing diversity of fungi that we find in British Columbia. Next, please. Next, please. So there are about 100,000 iNaturalist sequenced fungal specimens worldwide, representing about 7,200 different species. And most of these are from the 2 coasts of North America, east and west. Next, please. Of these, only 336 sequenced fungal species are from BC, based on 617 sequenced BC specimens. Next, please. So compare the BC number, for example, next please, to 1,738 sequenced fungal species in Washington. Next, please. 1,359 sequences, sequenced fungal species in Oregon. Next, please. And more than 2,500 sequenced fungal species in California. And all the while remembering that British Columbia is just a little bit larger than Washington, Oregon, and California put together. Right? So, in other words, we have only 2% of the number of sequenced INAT fungal specimens that are found in the 3 states in the US Pacific coast. And these states have a combined area that's smaller than BC. This anomaly cannot be explained by the ratio of INAT observations. BC has almost twice the total INAT fungal observations of either Washington or Oregon. And even throwing in the massive iNaturalist work from California, the headquarters of iNaturalist, the land area represented by BC has about 18% of the number of iNaturalist fungal observations found in the 3 US states. So we have not been sequencing nearly as many of our specimens as some of our other Western neighbors. This initiative that we're launching tonight is going to give us an opportunity to make some serious inroads into that. Next, please. If we look just in BC, we can see that all of the sequenced INAT species in BC are from the southern tier of the province. And in fact, 5% of the BC sequences are from Machosen, where Kim and I live. So that's rather a remarkable number. So we have a largely unexplored province overall, but the specimens that we do have sequenced are only from the very southern part of British Columbia, which leaves a huge area of the province unexplored, unsequenced. And so we're particularly pleased to have partners from northern British Columbia who we hope are going to be submitting a lot of specimens for sequencing. Next, please. Some sequencing results from the past 18 months here. I think highlight how much there is for us to learn. There's a lot of numbers here about sequences, failures, species confirmed, and so forth. I think I would direct your eyes to the lower right-hand side where the percent of total collections given a temporary code is 51%, suggesting that about half of the specimens that were coded on the screen here had to receive a temporary code because they were not a good match for described species that we had access to codes for. So we can expect to find a lot of new codes, probably a lot of new species in launching this MycoMap initiative. Next, please. Now, I just wanted to include one slide because a lot of what we're learning about how we were applying the wrong names to a lot of things can lead to despair. And I find that learning like this fills me with optimism. For many of these species we've known for a long time that we've been working with the wrong name. It's nice to see some confirmation of that. Learning the correct names for our local species can help us learn more about them and maybe how they differ from the species that we thought they were. And because our taxonomy and nomenclature, our classification and naming of these species are increasingly based on molecular data, they should be becoming more stable over time. So we live in a period of great change right now. It's a little bit confusing and frustrating at times. I think it's heading to a much better place for mycology and for everybody interested in fungi. Next, please. And over to Shannon.
Hello? Can you hear me? Yes.
Yeah, we can
Okay.
hear you.
Okay, thank you. Okay, so I'm just going to give you an example of the power of sequencing here in British Columbia. And my examples are based on a study that I had the pleasure to carry out in the Alpine, in some of the Alpine regions of the province, part of the province that is relatively poorly studied. And so this work I've done in collaboration with Anna Bazzicolupo and Ken Marr. So the Royal BC Museum had expeditions going up into alpine zones of the province for very, very many years. And they were focused primarily on documenting the plant species encountered, the insect species encountered, and describing the alpine ecosystem so that biogeoclimatic zone of the alpines could be improved. And one day, one of the people involved in these expeditions said to me, "Oh, and by the way, we see fungi up there, mushrooms fruiting all the time." So that gave me a chance to become involved in this project. And for 1 year, I was able to get up into the Alpine, assisted by helicopters, which was fabulous. Next, please. Okay, so during this project, we photographed all the mushroom specimens that we found, and we sampled tissue for ITS sequencing. So DNA sequencing specific to this zone called the ITS, which is used for fingerprinting the fungi. We did not do iNaturalist reports, so that's the big difference between this project and all the rest of the sequencing that's been done on fungi in the province. So here's an example. This is a Hebeloma alpinus, one of the fungi that we found, identified because of the ITS sequence that we got, but also morphologically confirmed by Henry Becker, who is a global expert of the genus Hebeloma. Next. Here's a photo of me hunched down in front of the lab tent up in the Alpine in July many years ago. And I'm sampling the mushrooms that we collected so that we can do DNA sequencing. So next. This is just showing you the vials that we used for preserving the fungal tissue. We took a little bit of gill tissue, individually put those into vials. In the vials, there was alcohol, and that was used to preserve the tissue until it got back to the lab. There's a different protocol for this particular study, but that's what we were doing at the time. Next. Okay, and then once we had the DNA sequences, we were able to explore the identity, the species identity of them in using the UNITE community. There's various ways of doing it, but I'm just showing this as an example. UNITE is a database and sequence management environment. It's based specifically on this ITS region, which is the focus of this IMAP project as well. And the UNITE community, it will cluster all of the sequences in the global databases to approximately the species level. And in UNITE, those are called species hypotheses. They have an SH number that designates that particular cluster of sequences. Now, and the fun thing about UNITE is that the user can interact with the species hypotheses in various ways. So if you're participating in this IMAP project down the road, you will have sequences of your fungi that you would be able to explore in UNITE. Next, please. Okay, so I'm just going to give you two examples of cool things we found. Here's two photographs of a collection of a mushroom. We ended up, based on the ITS sequence of these, we ended up finding two collections of the same species. And I call them permitted collections here because we were sampling in part in parks and protected areas. And so you have to have a permit to be sampling in there. So when we ran the DNA through UNITE, this is the result we got. Next. That this particular species is Amanita nivalis. And there's that very specific species hypothesis number. And the other fun thing I think that you can do in UNITE is you can see a map of where your collections, your sequences were located, and all of the other sequences in these global databases map throughout the world where the locations were. So in our case, there were our 2 collections from British Columbia, 1 collection from Greenland, 2 from northern Scandinavia, and 1 from Russia someplace. Next slide, please. Okay, and the other example I'm going to talk about is this little brown and slightly purplish mushroom. We only had 1 collection of it. It was from Mount Crysdale, which is west of Dawson Creek. And when we ran the ITS sequence through Unite next, this was identified as Cortinarius anomalous. And there again is that specific species hypothesis designation. And the map of the occurrences of sequences of this fungus that are clustered together in this species hypothesis. So again, you see our collection in British Columbia, there's some in Alaska, there's some in Svalbard, I think it's called, a Norwegian island up in the north there, China, and from Russia as well. So again, not very many sequences in the global databases of this particular taxon. So it's super cool to have a dot, I think, in British Columbia that links with all of the rest of these. Next. Now, the odd thing, an interesting thing in Unite is there are in fact 7 species hypotheses, all given this same name, Cortinarius anomalous. And next, that highlights for us that this is probably a species complex. People have applied the name Cortinarius anomalous. Somewhere in there may be the true Cortinarius anomalous, and perhaps there are 6 currently undescribed species, and ours may be currently may be an undescribed species. So again, the data that you're helping to generate will be used globally by taxonomists who are, as Andy said, doing their utmost best to help stabilize the identification and naming of macrofungal species. That's it. Thank you. Next.
Awesome. Well, thank you so much to Kim, Andy, and Shannon. That was great. And Stephen, I'll turn it over to you. You said you had a couple slides. Did you want to share?
Sure, if I have control, or you can advance it either way.
Oh, you threw them in here. Perfect.
Yeah, I just tossed them in.
Perfect.
Yeah, so, well, thank you all for having me here. It's great to see so many people. I see 65 participants in this chat at this moment in time. I've done a number of these types of introductions now. As you can see on the map, we've started projects in a number of places across North America. And this is definitely one of the best showings of local interest that I've seen thus far. And so it's really encouraging and really cool to have so many people interested in this type of work. So once again, my name is Steve Russell. I'm the director of MycoMap. It's a 501c3 nonprofit molecular research laboratory. We're based just out in Plymouth, Michigan, which is just outside of Ann Arbor, so near the University of Michigan. My day job is actually at the University of Michigan. Our lab is not affiliated with the university. It's a private, separate 501c3, but we do all of our sequencing work through the lab. And so I guess just to give a brief history of where all of this work came from. So I was actually going back into 2015, maybe 2016. I was one of the co-founders of the North American Mycoflora Project, which is now known as Fundus. So that would have been kind of the origin of how we started to get into this work and get into this philosophy. And if anyone's familiar with the North American Mycoflora Project, one of the core things that it wanted to achieve was to get DNA barcodes of all of the specimens, all of the species that exist in North America. And it's taken until 2025 to really kind of get the ball rolling in a really substantive way, to where we're starting to see massive gains year over year. And we're actually to a point now where we can kind of see that we can see an end game. whereas, you know, a decade ago, we had no concept for how much work we needed to do to kind of get towards the end. So we still have a huge amount of work left to do, a long way to go. And we're slowly opening up areas across the continental United States and Canada for these types of DNA sequencing initiatives. So Andy had mentioned several interesting statistics. Actually, let me stick here for one more second. The other primary project that currently exists in Canada is in Atlantic Canada. So the provinces that are highlighted there, they're currently sending in probably between 3,000 and 4,000 specimens a year. So you guys kind of have your work cut out for you to be able to catch up with what they're doing in Atlantic Canada. And let me tell you, the stuff that comes in from Atlantic Canada is some of the most interesting stuff that we've seen come into the project overall. I fully expect a lot of the stuff in the Northwest to be in a similar fashion. So in the US, there's a number of different hotspots. Next year, we're probably going to look to open up more states in the Northeast United States. So Maine, Vermont, New Hampshire. Probably looking to expand around the Gulf Coast a little bit with Mississippi, Louisiana. And that's probably going to be the extent of what we're looking to open up for next year. So we're just trying to take a gradual, manageable, sustainable process to where people from the highlighted areas can send in an unlimited number of specimens at any point throughout the year. And I'm very glad you guys can be a part of that. We want to advance one slide. So the MycoMap network with all of the states that we just saw is one of the core projects that our lab works on. The other one is something we've been doing since 2023 now, called the Continental MycoBlitz. And during this, we have 3 events throughout the year to where anybody in North America can submit specimens from anywhere. This year, we opened it up to an unlimited number of specimens. And this is really one of our other projects to where we're able to get, large amounts of mushrooms during the year, lots of people out collecting without being overwhelmed consistently. So our lab is actually pretty small. We have 3 people that currently work at the lab. So myself, a lab manager, and one other person that primarily works on culture projects. So it's not a large team that's able to do a lot of this work. One of the ways we've been able to continue to expand, in a cost-effective manner, is through really reducing the cost of sequencing, as Kim mentioned. Back in about 2022, 2023, we moved from a technology known as Sanger sequencing to a technology known as nanopore sequencing. And that dropped our cost per specimen by about an order of magnitude, which allowed us to process many, exponentially more specimens than we were previously able to do with Sanger for an even lower cost. And so that's really kind of the thing that's allowed us to continue expanding these projects substantially across North America. Let's go ahead and advance to the final slide. And as Andy mentioned, we are now over 100,000 sequence specimens for North America. Primarily, most of these, about 95,000, are from the US and Canada, with most of them coming from the continental United States, as you can see there. One of the things to keep in mind, I dropped a link in the chat, and Andy mentioned the temporary code names. The species counts on iNaturalist, it says 7,300 species listed there, but they don't currently incorporate any of the delimitations, any of the species that we've outlined that use these temporary code names. And so the real species count is significantly higher than that. One of the good bits of news that we just got this week, for those of you that are already integrated into iNat and understand how it works, these temporary code names are going to become a native part of their taxonomy here probably in the next few months. So they had a generous contribution, I believe, from somebody in the mycological community to allow the integration of these temporary code names into the native framework. And that's going to make our work exponentially easier. It's going to make INAT's computer vision way, way better, because it's going to allow for better clusters of the morphology of these things. And so that's something I would say that was the number one thing on our iNaturalist wish list, was to be able to incorporate provisional names into the core taxonomic framework. And that'll really help to get the species counts more accurate when we're doing searches like this. And so I think I'll leave it there for now. I'm definitely going to stick around to answer any questions that you guys might have. But I'm really excited to have you all a part of this, and look forward to this project getting kicked off.
Awesome. Well, thank you so much, Stephen. I realized I didn't really, well, you introduced yourself, and that's great. And maybe just to make it clear, if it isn't already for folks, that Stephen's going to be processing all of our samples. So I mentioned earlier, we have that Google form. When you fill it out, you're going to get an address. Basically, your specimens are going to be sent to me, but I am just a muster point for your specimens. And at that point, I'll hang on to them until we have a nice batch, and then they'll be sent down to Stephen. There's not a ton of people. Well, there's very few people. In fact, maybe there is currently 2 people doing such high throughput nanopore sequencing of fungi right now. So it's a pretty niche service that Stephen offers, and we're really, grateful that we can get BC joining the MycoMap network. Yeah.
Yeah, I just mentioned that's the aggregation, the local aggregation of these specimens is really one of the things that helps us out the most. The thing that takes us the longest at this point in time isn't the DNA sequencing or cost, right? The thing that costs us the most at this point is the time that it takes to organize specimens and to take tissue. And so, if we can have that local triage, a local resource to where specimens can be sent for that preliminary triage, to give them that initial sort and organization, that really saves us a lot of time at our lab with our limited staff to allow us to make much more progress. And so that's actually one of the reasons why some of these other areas where there are lots of collectors that we haven't opened up yet, because we would just get overwhelmed with way too many specimens. And so really having this locally organized project is going to be essential to success.
Awesome. And you have a lab member from BC, don't you? One of your staff?
Yeah.
Just a fun fact.
Yeah, for a period of time, yes.
Okay. Sweet. Okay, well, that brings us to diving into the project protocol a little bit more, and Anne's gonna take it away with this. So I'll keep my screen share on for a second here, Anne, and then just let me know when you want me to turn it off, and you can take over.
Excellent. Thank you so much, Alora, and thank you so much, Stephen, for joining us this evening and explaining a little bit about the really important work that Mycota Lab does. We are so grateful for your visionary approach to mycology and the ways that you are pushing boundaries with taxonomy, and it's really cool to join the MycoMap network and get to be a part of that. I would also just like to highlight Elora because she has been by far the biggest force behind getting this project off the ground. She has done everything all the way down to the drawings of the mushrooms on the different resources that we've put out for the public. So, she's been the true force behind this project. And we're really grateful to have that, because there's no way this would have all come together without Elora's involvement. So tonight I'm going to be helping you guys, especially folks who are newer to collecting or newer to mycology. I'm going to be helping you understand a little bit about our collection protocol and how you can participate. And so if you've never done this kind of work before, that's okay. I'll give you a little bit of an introduction, and then we have a full protocol that lays out everything in a lot of detail if you're a details kind of person. Next, please. So to start, these are some beautiful graphics also made by Elora with cute little mushroom drawings. There's kind of, 4 basic steps that you're going to go through when you're participating in this initiative. So first is find a mushroom. Then you're going to dry that mushroom You're going to upload an observation of that mushroom to iNaturalist, and then you're going to bag that mushroom up and send it off to us for that preliminary triage, as it was put. So 4 main steps there. When you do find a mushroom, you're going to want to take some photos of that in the field. For this, you can either just take the photos directly on your phone, or you can do it directly in the iNaturalist app. Everyone has kind of a different approach to this, so find what works for you. But the most important thing is that you're capturing photos that show all parts of the mushroom. So you want to be able to see the different features, including the base of the mushroom. So it's important to carefully excavate that mushroom and make sure you photograph all those details. We are also providing, through Stephen and his excellent work, some field slips that you can use to make vouchers in the field. And so you'll have a little slip that looks like this, and I'm actually using a Continental MycoBlitz one tonight, but ours will have the little BC 25 for our British Columbia specific project. And you can use this to fill out some information about the mushroom that you're able to observe while you're in the field. And I do recommend to do that while you're out in the field, while that information is readily available, because you'll be capturing notes about things like what kind of trees are growing around, or what kind of substrate the mushroom is growing from. Is it growing off a log? Is it growing off the soil? Is it growing on a pine cone? There's many different ways that you might find that. And so you'll try to fill out as much of that information as possible. And then at the bottom of the little field slip, there's a nice little tag that you can tear off. And I kind of pre-cut mine, so it's easier to rip this off in the field, and I'm not destroying my field slip while I'm there. So I just kind of go through and I pre-cut them before I take them out. But you'll keep that little voucher label with this unique ID with that mushroom throughout the whole thing. So you can keep track of what mushroom that goes to and what iNaturalist observation that's going to go to. So another important component is, as you can kind of see on the step 1 image there, it's a good idea to get a picture of your mushroom with that voucher slip there that shows the number, just to make sure you can cross-reference that later. So then you're going to, well, okay, before we get to drying the mushroom, I'll talk a little bit about collecting and different tools for doing that. So your field collection kit, you're going to want to have some kind of container that you can collect your mushrooms. Personally, I like to use a little plastic tackle box I just get mine at the dollar store. You can get it there, Canadian Tire, a bunch of places. You could get them online. There's different styles. This one is like a fixed size, so all the little compartments are the same size. I use this one for collecting smaller mushrooms. And then there's all sorts of sizes you can get. Here's a bit of a bigger one that I use, and it has removable dividers, so I can kind of customize if I'm going to have a larger mushroom, things like that. And what I would recommend, though, you'll notice on the back here, the hinge, this is just one of those folded plastic hinges. I don't particularly like that because they wear out quite quickly. So I would recommend finding a collection box, if you plan to go this route, that has an actual pivoting hinge, not one that's just like a folded plastic hinge. And this will hold up a lot better over time. Also, you can kind of see with the latches here, kind of the actual turning of that mechanism. So I would recommend that. Some people also will just do wax paper. You can wrap your mushroom up in kind of a bonbon style with a piece of wax paper and make sure that voucher slip's included in there, and you can keep them all organized that way. And if you have your tackle box, it's also important to keep that little voucher tag with your mushroom there as well. So once you're ready to collect your mushroom, you've gotten all your pictures and you've excavated it and captured all the different angles and everything. You're going to take that mushroom, tear off your little voucher tag, and then your mushroom, here's my little example mushroom here, the Strobilurus trillatus from the Doug Fir cone. You're going to want to remove any substrate that you have with that mushroom after you've gotten all the photos of that. And that just keeps everything a lot cleaner and makes it a lot easier when you're futzing with this all later. So you can remove your mushrooms from the substrate. It's a really good idea to carry a nice little knife with you out in the field so you can trim any kind of debris or anything off of the mushroom there as well. And then you can place that mushroom in your handy little collection box, like so, with your little voucher slip, just trying to keep everything organized, like there. And then you can make a bunch of different collections while you're out, but don't feel compelled to capture every single thing you find. The first time I ever participated in a MycoBlitz, I sent in a grand total of 4 mushrooms. So don't feel like you have to send in hundreds of things. Feel free to just start participating in whatever way feels comfortable for you. So then the next thing is you want to get that mushroom home and get it dried as soon as possible to prevent any degradation of the DNA. You don't want them to turn into a big ball of slime, which is another good reason. I mentioned wax paper is a good way to do. These plastic little boxes are good too. Don't use plastic bags for collecting because they will turn into balls of slime and it is not good. So no plastic bags for collecting in the field. Then you're going to bring home your mushroom and you're going to get that in the dehydrator as quickly as possible. If you already have a dehydrator at home, that's great. You can use that. I would probably wash those racks and stuff if you're going to be using that for food later, just in case you have any cross-contamination from a poisonous mushroom. It's not a huge area of concern, but just basic safety would be good there. If you don't have a dehydrator, you can pick one up pretty cheaply at a thrift store or on Facebook Marketplace, or you can borrow one from your roommate, which I have done. I've borrowed the dehydrator here, and it's just one of those old style type things. And Laura, I think actually, if you want to kill the screen share, since I'm gonna be doing more like showing instead of the screen, that's probably better. Thank you. Yeah, so I just use one of these dehydrators. This thing's honestly probably about as old as I am. But it's just one of these top-down ones. It blows warm air down. It has a little temperature control knob on here. And I recommend to dry your mushrooms at a low temperature so you don't completely fry them, but you do want them to be completely dry. So this style dehydrator, if you're not familiar, just kind of has these racks, and so you'll fill these racks with your mushrooms. Again, making sure to keep that voucher tag with the mushroom. And this is a place where I have struggled at times because sometimes the little mushrooms will get blown around in the dehydrator and separated from their voucher tags. So when I go to put that little tag with my little mushroom, it's a good idea, especially for little small things, to use a bottle cap or something like that. And you can place your mushroom in the bottle cap. And then in this case, I'd probably just put the bottle cap on top of the slip and then place it on your dehydrator rack so you can dry that. If you have a bigger mushroom, then you can just place that mushroom on top of the voucher tag and that will keep it together. And a good note too about large mushrooms, especially if you have like an amanita or a large russula or something like that, you don't need very much of the mushroom. So don't feel like you need to wait a million hours for a massive mushroom to dry. Even in the field, you don't have to collect the whole mushroom. If it's a big mushroom, just take something like a quarter of the cap and bring that home with you, and that will be plenty of tissue to be able to use for sequencing and sampling and everything. So generally we say dry your mushroom for 12 to 24 hours, but the real test is when it is cracker dry. So if you were to bend it, it is going to snap. There's not going to be any give or play or flexibility in that. And then when you're checking on your mushrooms to test, like see if they're pretty dry, one thing I do recommend is turning off the dehydrator before you open it up. I learned that the hard way when I did that once with the dehydrator still running, and it blew all my tags and all my mushrooms everywhere, and it took me a second to go figure out where everything went. So once those mushrooms are completely dry, then you're going to bag them up and put them with your voucher slip. So what we recommend and what works really well for Mycota, because they're going to be storing all these things in their herbarium, we want to take up the smallest amount of room possible. So I like to use the snack size bags, and you can, like a Ziploc snack size bags. Those are good. So yeah, once those mushrooms are dry, you're gonna put each one in a bag with the corresponding slip. And if you trim the edges of these little voucher slips all the way down, it'll fit in that bag, and that's really nice. So that would be the preferred way of doing that, is making sure that that slip is included in the bag with the mushroom. Another important thing that you'll do is when you go and you make all of your iNaturalist observations, whether you're doing that in the field or if you're uploading things later at home, is you want to make sure that iNaturalist number is included with the information that you're including with the mushroom. So there is a little field at the bottom of the slip for the iNaturalist number. Please make sure that you fill that out, or we won't be able to cross-reference it. And that's a very important thing for the uploading later on. So when the DNA results come back, that's all going to get uploaded to iNaturalist and linked with your observation. So make sure that you can link back to that and find that information. The other thing too, in terms of iNaturalist, is we want to make sure that we add the observations to the project. So I'm actually going to share my screen and just show you real quick an example of that. All right. Okay, so in iNaturalist, here's my observation for that mushroom that we've been working with here. And the first thing you'll have to do in order to be able to add your mushroom to the project is to go and join that project. So you can search for the MycoMap BC project. And it should come up here, the little blue background with the mushroom, MycoMap BC, a DNA sequencing project. So you can go in that, and then there will be an option. I think it's usually in the upper right-hand corner somewhere to join that. So once you've joined, you can go to your observation and you can add that to it. It is a manual adding. We don't have like a geofence or anything to add things because we want to make sure that we're getting the right collections and things. So you'll scroll down to projects and then you'll find it should be in your list, or you can type it in if you don't see it because you might have a lot. We'll add it there. And in order to add to the project, you will have to select that, yes, you have taken a voucher specimen. That's that mushroom that you're sending in. And then this is a really important point, and it's really annoying. Usually I just click yes, and I try to click add to project, and it won't let you. You have to click this little add button here first, for whatever reason. It's just part of the interface. Click that first, and then you can click add to project. And then you should see that MycoMap BC has been added there. So that's kind of the basic overview about collecting and adding things to the project. You guys are going to be able to find a lot more information and things of that nature through that MycoMap BC. So if we go to mycomapbc.org, there is a link here for our Read Our Protocol, and that's going to take you to our Google Doc here that has all the information. What do you want to do before you go into the field in terms of setting up iNaturalist? Here's our recommendations for how to put together a field kit, the different supplies you might need, and then a nice little collection procedure that helps walk you through those steps of how to pick good mushrooms for this. You don't want to pick old, gross ones. You want to try to find something that's fresh and doesn't show signs of significant decay. Unless it's something you think is going to be like really, really interesting and it's the only one around, we can still try, but it may not come through. So where possible, please do go for the freshest ones that you can. So that'll just go through all that. It talks about all these different things we've kind of run through in a high level, and this gives you a little bit more detail about that. And then, of course, the transportation, dehydrating, and all that kind of stuff. And if you're really interested, you can send a split of your mushroom to an herbarium if you wanted to document that and have that locally preserved. It'll also be preserved in the herbarium or fungarium at Mycota Lab, but if you wanted to have that either in your own personal collection or to send that to somewhere in BC, there are some options for that, and we've put together a little bit of a list that explains different herbaria in the different regions of BC. Not all of them are accepting things from the public, but you can always check with your local herbarium and see if they are and how you could contribute to their collection. All right, so I think that's pretty much what I had in mind to go over. And I know we do have a lot of experienced collectors on the call. So if anyone else has anything they would like to contribute or mention or tips that they have found helpful when they're doing their own collections, I would invite you to chime in.
And there's just been a couple things in the comment or in the chat. A lot of them have been answered already, which is great. So if you don't have the chat open, there's been some discussion there that's helpful. One thing that I thought was good was point out how to find the INAT number. So if you can just actually start your screen share
Yes,
again and just show, or I can just tell people the
I got
easiest.
it.
Yeah, sweet.
Yeah. So yeah, the one thing too that I forgot to mention when you're adding to the project, at least on iOS, there's no option to do project things and fill out those fields on the app. So you will have to go to either the web version or to an actual computer to do that part. And same kind of goes for finding that INAT number. It's basically just the tag that is part of the URL. It's that last part of the URL, those digits there. That's your iNaturalist number that you're going to put on your field slip, and that's going to relate that back to your mushroom.
You can see the number in the new app quite easily.
Oh, good.
Yeah, on iPhone at least. I don't know about other phones. But yeah, you can do all your uploads from your phone still if you'd like. It's just this one part about adding to the projects because it's not an automatic add that, yeah, it's a manual step that has to happen on the computer. Um, let's see. What was the other question? The voucher number field. Yeah, so there's two fields that I add when I upload a mushroom that I've vouchered for sequencing. It's the did you take a voucher? Yes, and showed you that one. The other one is voucher number. So there's a few that say that voucher number with a S in brackets, just something that you can add that unique code from the slip into the observation. That's the second step that's quite helpful, is adding that as well. That is not necessary as a field to add it to the MycoMap project. Maybe I should make it necessary, but yes, adding that to easily link the two things would be ideal. These questions have been answered. Yeah, no lichen, slime molds.
This is Kim here. I would also mention a step that I take in between what Ann is doing. That is, when I collect a mushroom and dry it, it goes to my own personal herbarium. And I try to get the whole mushroom there because there may be morphological aspects of it that need to be examined at a later point. And then from my own collection, then I go to the stages Ann was talking about to prepare it to send in. And when you put a piece of it into the bag, you just need a very small piece of the mushroom. And the rest of it would remain in my own personal herbarium that someday I hope would be transferred to a public herbarium somewhere.
Yeah,
That's excellent.
and that's a great point because in Atlantic Canada, they have Alfredo Justo, who is taking splits himself before they go to Steven. But yeah, for us, just because of the amount of work it would take to get all those mushrooms in for like me or Anne or someone to take those splits, we're just asking folks to send a small piece. And then, yeah, if you do have the interest and the capacity, saving the rest of the specimen and in your own herbaria is great. Otherwise, the rest of it will be saved at Mycota, which is a good as an alternative. Okay, I see Bill has his hand raised. Why don't we, Bill, go ahead and then we'll move along because we will have a bit more time for questions right at the end.
So it's a quick little one. Today I collected a piece of some slime mold, but when I went to pry it off, it just like dissolved into dust, which I have collected. Is it okay to send like a little foil pouch? Like I put it in foil and folded it all up because it's just like dust because it's like one of the dog vomits, you know, when they look like a fried egg.
Yeah, actually, I'm going to defer to Stephen on that because, yeah, people do send things in all sorts of manners, but it'd be good for him to chime in on how he would best like to see that.
Yeah, so just this year, we decided to start accepting slime molds. I would say we don't have a mature protocol yet for slime molds, but we're working on that. And so we're starting to allow people to send in collections. We'll probably only do, so it's going to require a special workflow, and not all slime molds use the same workflow. And so they're a little bit more difficult to deal with overall. But that's definitely something that we're in the process. So we accept them currently, and we're in the process of getting some, solid workflows together for slime molds as well.
So the slime, can we just leave the substrate on with the mold?
As of now, I think that's appropriate, just because they're difficult to deal with. Like I said, we'll probably have more guidance as we start to see, how many hundreds of different slime molds come in and the condition and what kind of issues we have with sampling them. But for now, yes, I think a small bit of substrate attached to it is perfectly acceptable.
Awesome. Okay. I am going to pull up a last couple slides here, and then we'll have time for a few more questions before we wrap up, if that sounds good. So, just wanted to highlight a couple upcoming things. Many of you probably are well aware of all the fungi events happening in the province over the next few months. But the first one here is kind of our own little online mycoblitz. So, I can't exactly remember last time I looked at our project on INET. We do have like 43 people in the project. And do you mind sending a link to the project to the chat so anyone who already has an iNaturalist account hopefully can join that project if you haven't already, and then start adding your observations. But we picked this sort of arbitrary week starting tomorrow, September 17th to 24th, with the goal of hitting our first 1,000 observations. We already have close to 500 in there, I believe. And we just picked this week because by the time October rolls around, like things are crazy. There's a million other fungi events happening, and this would be just totally lost. So what we're hoping is that, folks can feel motivated to go out there over the next week and contribute as many observations and collections as you'd like. My goal is to, convert maybe casual mushroom appreciators into people who, carry around a tackle box, carry around a couple slips, and when they see something that catches their eye, or maybe they know it's different, they've never seen it on this trail they frequent, they'll make a collection. And also to convert plant people, bug people, all you other people into people who would make a couple mushroom collections when you're out there doing your own thing. So this week is just like a good week to start that off, to go out there and try to get our numbers up on iNaturalist. I have a crazy goal of us getting 10,000 collections in the next 12 months. I was told that it was a little ambitious, but I kind of think it could be possible. That's like 200 people making 50 collections, or rather 50 people making 200 collections might be more appropriate. But anyway, if you are keen, we'd love to see the numbers going up next week. There are also lots of events at which either myself or Anne or Andy or Shannon or Kim or there's other folks out there, Aaron, Juliet, are going to be running walks, running talks, and we'll be talking about MycoMap, providing like in-person sort of tutorials on making collections, speaking more broadly to the project as we did tonight. So if those events listed there are anywhere near you, you can come out and sort of have a deeper dive, get a chance to make some collections in the field. If you are making collections and you have any very impressive mushroom finds and some nice pictures, feel free to tag or invite MycoMap BC on Instagram to be a collaborator. And if you want to host a training for your mushroom club or for your region, get in touch. Maybe I don't know if my email and Anne's email are on the MycoMap website, so you can find it there. As I'm gonna say, especially Northern BC, I would love to get some things going up there, run a training for more folks from the club, because we didn't see that many Northern BC people when we did our poll, or to provide the resources for someone up there who could totally like run this kind of training on their own. So yeah, if you're part of a group and you think that this would be of interest, we'd be happy to make that possible, so that more people kind of know what's up and how to participate. Cool.
Hey, Laura.
Yeah.
I just wanted to add a couple things. Cam had reminded us, what's the address to send collections to? So, real quick, just to follow up on that, I'm going to steal the screen share for a second. When you guys do have your mushrooms and you're ready to send them off, you can go back to the where's our MycoMap BC page? Gotta pull that up again. If you go here, there is a button here for ready to send your specimens. If you do that, it'll take you to this little Google form, which will just help you go through and make sure that you've done all the steps that we're asking to make sure that you are providing good, useful collections. And when you fill that out at the end, you'll get the address for the lab where we'll be sending everything as the muster point here in BC. And then also, Laura, I'm going to hand it back over to you to wrap up here in a moment. But I did just want to say that I forgot to mention at the beginning, that I'm just really excited about this as a citizen science initiative and what I believe is probably the largest mushroom-based citizen science initiative to ever happen in BC. And it really provides a chance for anyone to be able to contribute to the really important discoveries that we're making. And I just think that that's a really cool thing. It's never really been possible before. And I hope everyone is excited and wants to get out there and find mushrooms and help for science.
Awesome. Yeah, and there's been a couple other great questions in the chat. This was something I meant to answer, but if you have specimens from 2025, we can accept anything from 2025 onwards at this point. So yeah, we'll keep going, you know, throughout the fall, but if you have things that have been sitting around, those can come in as well. So that is like sort of the official start date. Sweet. Well, that brings us to the end. And we can have a couple minutes here to answer any last questions. So for folks that maybe want to head out, I know we've been on Zoom a while here. I'm just going to echo Anne. I'm excited and I feel very ambitious about actually how much collections we can get from the province. Fungi season is sort of just going to be starting soon on the coast, and yeah, it seems like there's a lot of interest, and I hope we can start to catch up to our southern neighbors and impress all these other MycoMap projects with how much stuff comes in from BC this year. Yeah. Okay, Q&A. So if you need to head out now, thanks so much for coming. This recording will be available. I see a request for the slides. I can send out the slides as well. I have a list of emails from everyone who attended, so I'll get that info out with some maybe like pertinent links and things that were discussed tonight. Yeah, so if you've got to go, have a good night, and otherwise, feel free to ask your questions in the chat or maybe use the hand raising feature, and we'll try our best to answer them.
And Laura, where do we get the forms to fill in?
Oh yeah, so the little voucher slips, the field slips.
Yep.
Yeah, so those are on the website. If you go to mycomapbc.org, you will see a link and then you can generate your page of field slips. It will create the unique numbers from you, and then you can just print off as many as you'd like. And if you don't have a printer, go to the public library and you can print for free.
Am I live?
Yeah.
Okay. In Colombia, it's illegal to send any biological material out of the country. Do you know if there is such a restriction to send material from out of Canada?
Nope.
You have to have a permit to get to
I
send
mean,
it.
I can grab that one, Laura.
Yeah.
Yeah, so no, it's okay. What we'll probably be doing, like, and this is a point, part of the reason why we have one central point to collect everything before it gets sent. But usually what I do is I just fill out a customs declaration that references the
Yeah.
specific part of the code that deals with dried, preserved museum specimens for research purposes, and that gets it through. But I've sent things to Steve a bunch of times for Michael Blitzes, and I've never had a problem.
Yeah, myself as well.
Yeah, it's just a matter of declaring everything properly.
Sometimes the people at Canada Post are like definitely looking at you funny. They're like, "What is this?" And it's a
Yeah.
bit of a conversation. It's a conversation starter, you know, in a positive way. So, but yeah, I haven't never had any problems.
It's
Yeah,
actually not a question of whether the postal system will handle it or catch it. It's actually in Colombia, it's actually illegal. And you can, there are very serious fines if you are caught sending stuff out. So it's a question of law.
It's a good thing we're in British Columbia.
yeah. There's certain countries around the globe that don't want their biological material exported, and so it's a local regulation against export. There's no U.S. restrictions against import for those kind of things. We get packages from all around the world regularly of fungi. It's only been stopped by our customs once,
Mm.
and that's because they thought it was lichens. And when I told them that it was mushrooms, they let it right through. So it's really mostly they're concerned with plants and soil, much more so than they are mushrooms. But any country where it's legal to export mushrooms from the country, we can accept them anywhere globally. But that's in Colombia, it's probably more of a local restriction, a local law.
Thanks, Stephen. Okay, I'm just looking in the chat here. There's a question about an air fryer dehydrator setting. So I'm not really familiar with what air fryer dehydrator settings would be like. Basically, we're looking for something 60 degrees Celsius or lower. Like I usually drive mine just around 50 actually. Someone asked about an oven earlier. Ovens typically don't have a setting that low. If your oven does, then give it a shot. But like my oven doesn't run lower than like 200 or something. Well, anyway, it doesn't run as low as 60 degrees Celsius. So if it seems like it does, then I think it's probably worth a try. Otherwise, yeah, I would try to track one down for cheap on Marketplace or at a thrift store or something, or they're not that expensive new either. Yeah, any other questions? If questions are coming up later, yeah, find Anne or I, or ideally both of our contacts on the MycoMap BC website and shoot us an email for sure.
It's still weird here. Is hiding the location okay? Because I want to do a bit of a sample off an agaricon, but I don't want it to be known. Like
I
it's
feel,
BC.
yeah, I don't imagine obscured location would cause any problems.
It's also possible to share the location only with the individual.
Trust the project coordinators. Yep. Yeah. You'll be given when you join the project bill, it'll give you
I'm in
question.
already.
Okay, well then you would have already selected if you wanted to. If you wanted to share the hidden coordinates with project
I'll
coordinators.
just put a big circle.
Sure, yeah. Yeah, gotcha. Yeah, and then I'm seeing the question about collecting in parks. So no, you can't collect in provincial parks without appropriate permits. So ways to get around things like that, or not get around things like that, but find opportunities to collect in parks or to join groups that do have permissions to collect in those parks. So the Mycological Society, for example, when we go out to CRD parks or we go out to like certain private properties at universities and such, and we have agreements and permissions to make collections in those areas. So if you're in the Mycological Society here on Vancouver Island, I'd recommend coming on some of our walks, in which case those permissions would extend to you, or looking into local clubs and seeing what permissions they have to collect. I know Vale has been doing some work and research compiling information on how to get permitting to other places. Vale, do you have any comment you want to add there?
Yeah, I'll put together a list. It's not that difficult. I've found talking with park rangers at different places seems to be okay, but I haven't looked into the actual regulations for provincial parks. I have acquired national parks and Nature Conservancy of Canada, and yeah, I've heard that we can do CRD if you're a SWIMS member. But there's a few other things that I could put together something, or I'm working on putting together. Just some protocols. It's not difficult to get permission in most cases, so it's something to look into, not try and skirt around the rules.
Yeah, definitely. And if you come up with a list, I'm sure folks would appreciate seeing it. And yeah, what Kem said too, when I was saying that those permits would apply to you, they'd apply to you when you're on a SFIMS walk. So it's always worth, what I'll add, it's always worth reaching out. If there's a place you'd like to go collect that you think has interesting things, be it a park, a municipal park, a provincial park, private property, conservancy land, it is always worth sending an email to the appropriate body explaining the purpose of your trip and your desire to collect, and seeing what can be done. There's properties SFIMS has gained access to that are typically not open to the public, but they've brought us in because they too are interested in the data that can come from bringing in mycologists to that area and collecting and sampling and sending things for DNA sequencing. There's been provincial parks that SFIMS has gained access as an organization to collect at during our forays by reaching out to the appropriate park supervisors and setting that up. So especially if you have any sort of like formal body, be it a mycological society or an other naturalist group or affiliations with a university, perhaps, I think leaning on those things and then simply asking for permission sometimes can be pretty successful for this type of project, because often those, the groups that are responsible for that land might be equally interested in getting this data. Like it could be a mutually beneficial situation. So yeah, don't let that hold you back. Try to, if there's a place you're interested, I think reaching out and seeing what's possible.
Yeah, with I'll just add a couple things. Yeah, with mushrooms especially, the way that I've approached it is that they're very neglected in what kind of organisms are in different property descriptions and stuff. And I've received nothing but positive feedback. And yeah, we'd love to find out more, especially when you say, you found some new species already. People are very interested in finding out what's on different properties. That goes right up to the national parks. They were really enthusiastic about finding out more. There's just not anybody doing it. So this project's going to really open up some doors.
Yeah, to answer the question about how long does the project run for, initially we'll be running it for the next 12 months, and then we'll touch base from there with hope to extend into the future for sure. Awesome. Well, I feel like we've touched on most things here. We've had some great questions. We've been going for about an hour and a half, which was what I was hoping for actually. So that's great. So maybe it's a good time to wrap up. And again, check out the website, the social media, and just shoot us an email. I will try to be a fast email responder and get you some answers if you have any. And yeah, if you're part of a group, especially in an area of the province that's undersampled, I would love to talk more about how we can bring this sort of like training or some version of it to people in your area. And yeah, that's it, I think. Thanks so much everyone for being here. Can't wait to see what kind of mushrooms you find. All right.
Thank you, Laura.
Yeah. Okay,
Thanks,
have a good night,
Alora,
everyone.
and speakers.
Okay, I'm
Thanks,
ending the
everyone.
meeting.
Bye-bye.
Take care.
Thank you